It's such an honour to be here at such short notice. And thank you everyone in New Zealand, Auckland and Wellington, of being so welcoming to me. I could never have believed that this would have happened. So what I'm going to talk to you today about is, the title's Exhibiting Oceans and Changes in Museums' Collections. I'm going to take you through a broad brush of what I've actually been doing over the last 30 years. I should have been doing something, right? And how things have changed in terms of how we study the specimens, how we do science now, and the ways forward. And along the way, how certain narratives are being more included now, and museums being more inclusive, and in terms of the staff and the stories that they tell. So this is a slide that some of you who have been following me online have seen before, but it's a beautiful image of showing you what historical fields collection looked like in the 1830s. So not just European naturalists, but also people of colour. And this is a painting by French artists who went to Brazil. So you mentioned Brazil at the beginning. In the 1830s, mid-1830s, now I've forgotten his name, Jean Baptiste Debret. And he was documenting life there. And this is a beautiful way and how we can actually use historical paintings to look how what the environment was like and compare it to how environments have changed, the landscape as well. Not just about how beautiful it is, but what is actually going on and the different plants and animals there. But also this is showing the different animals that were collected that are actually housed in natural history museums around the world there. So it's just giving you a flavour, a broad brush of all the types of collecting and who was collecting and acknowledging that as well. So I'm part of Modern Field Collectors. So I'm actually a marine biologist, but you can see I've got a net in my hand, a different kind of net. I'm there with my colleagues and some volunteers. We're actually at a place called Broad Point, where we're actually collecting insects. So I do help out and do that. So I'm a marine invertebrate expert, but also a generalist in terrestrial invertebrates too, so collecting insects there. But what does that mean to be a modern field collector? Well, it can often mean this for me. So this is what life looks like in my lab. And for those of you that aren't scientists or generalists in the field, the jars that you can see that are painted red are actually known as type specimens. So they are the name-bearing species housed in those jars. So the first of any species named found anywhere in the world. And so the Natural History Museum, as well as many across the world, but in the UK is a very large worldwide repository of type specimens. So this is where scientists will come and actually compare their samples, and they actually can loan them as well. So we do scientific loans where we package the material and it can be sent to scientists in their lab for comparison of work. And you can see that I'm there handling with my gloves on a very large lobster. So everything in the lab there is preserved in 80% ethanol, or alcohol as we call it, preserving material for forever, essentially. So I don't spend that much time, you can see I'm here, I don't spend that much time looking down at a microscope these days because of the length of service. You move higher up the hierarchy within an organisation and are now often supervising PhD students in their studies and probably doing additional studies that I've always wanted to do. But it's nice to give them a sense of direction and work alongside them and guide them in their work. But I do, when I get back home from this, I have some samples to look at from a British overseas territory. But just to give those who don't know what crustaceans are or might not be used to the variety of them, so here's some pretty pictures of them. So actually there is one animal at the bottom in the centre, which is, what would you call it here, landhopper, is that right? Yeah, locally, the common names you see. So this Articelytrus dorenii originates from Australia, this side of the world, and came in at the time as an invasive species on tree ferns. So when the Victorians had a passion for tree ferns and came in the soil there, and has become abundant in lots of parks and gardens across the UK, in particular in London, and first observed in the soil in a greenhouse at Kew Gardens in 1981. And I spent a lot of time, literally 25 years, monitoring them in the wildlife garden at the Natural History Museum as well. Not sure what you would call these locally, but they're called woodlice for me. So they're terrestrial, so there is a little bit of a terrestrial arm to the research that I do, and people find them in their gardens in the UK. And just some others, the Chinese mitten crab, and some other spider crabs, and also a blue swimming crab. So I look after, so the things you see in jars are known as wet collections. So anything from the spiny lobsters that you can see on your right-hand side and to dry corals. So corals are cousins or related to jellyfish as well. I look after that collection too. The interesting thing about these two specimens, you sort of think, well, taxonomically or in science, they have no connection whatsoever. But historically, they do, quite interestingly. And it was only during the pandemic that I actually had a eureka moment where I was working in a storeroom with the coral collection on my own, which was collected on an expedition in 1924, the East African expedition. I keep wanting to say exhibition, tongue twister. But then on the lobsters, you can see on the label, it says Dr. Leakey. And I thought, oh, I only know Dr. Leakey or the Leakey family from anthropology. And I thought, oh, that's a bit strange, what they're doing with lobsters, Kenya. But it turns out that both the coral and the lobsters are then connected to the main scientific work on that expedition in 1924, where Louis Leakey, he was around the age of 21. He went on this expedition to West Africa to actually help digging for dinosaurs. So dinosaurs is a connection of these two lots of samples. And what I have then tried to do is to link through our scientific database at the museum these two lots of specimens and the dinosaur data, and actually the West African people that were also collecting dinosaurs as part of this whole expedition process, to get everything together holistically. Because at the moment, the way that we do science within natural history, it's one bit over here in paleontology, one in zoology and whatever, in terms of our scientific disciplines and the way we work. But databases and data, environmental data, and who collected them can be joined together in a more holistic way. It's not perfect, but we're getting there, and people like me are one of the voices that are trying to do that kind of work. So I mentioned in one of my pictures earlier on about the Chinese mitten crab. So in the UK, in the top 10 of invasive species, 5 of them, so more than half of them, are crustaceans, and so crabs, shrimps and lobsters are in that. And we've got the Chinese mitten crab. Mitten crab, because it has furry claws, it's debatable whether you consider that hair or fur. But what can be found in them are tiny shrimpy things called amphipods. So that was a landhopper, a similar animal to the one that I showed you earlier on. And it seems that they live in the hairs of the mitten crab to gather small particles of food once the crab is feeding. They're an issue in the UK because their larvae has come on the ballast water. So through economic situations of goods sort of coming from Southeast Asia, China, places like that, the ballast water, that the larvae of the crabs get turned around in that water and then churned out when they land in places like UK waters. That's why it's very important for, in terms of any kind of crab larvae, barnacles that adhere themselves to the bottom of boats and ships. Well, actually, you need to have clean gear when you're going through the waters. I, interestingly, 30 odd years ago, when I was a junior grade, manned the mitten crab hotline. And when we were using ordinary landline phones, no mobiles then, dare I say, or no internet then, because I joined the museum in 1991. And the whole point of that was to take calls and to document on the map of the UK on the various rivers that stem from the River Thames where these animals were being found. It did end up in a bit of a situation from the anglers, so the people that are doing fishing locally, that they weren't getting any fish. They weren't finding any eels, the native eel population, but what they were getting on the end of their lines were the mitten crabs, and they were sending them into the museum. We then got to a situation where we had to say, no, we don't want any more mitten crabs, thank you. Just tell us the locations. That's what we wanted with the environmental data. So stemming sort of going on forward to 30 years, we're still studying the population. And they are a threat to our native crayfish and eels, and also to the bank sides of any river, in particular the River Thames, because for breeding they tend to burrow in there and uproot the vegetation and can cause potential landslides. And so we are monitoring their situations. Oh, I can hear that I've come out. Right, okay. So over the last 30 years, we've been monitoring their populations, and I've had sort of architects looking at whether they could design structures that could stabilise the bankside of any river, but also give a home to these mitten crabs, because I think they're here to stay. So I mentioned barnacles, and they, can be such a problem to the shipping industry, causing drag if they're encrusting on the underside of boats and ships, which would then mean economically you would have to use more fuel to transport goods. So this collection you can see here, I've chose the most glamorous of barnacles. So these are stalked barnacles. Some of them still have a bit of colour in, amazingly. And so this is part of the collection that I look after, which is the Charles Darwin accessed in the 1840s. So he literally persuaded the British Museum. So at the time in the 1840s, all of the natural history collections were still housed at the British Museum. It wasn't until 1881 that the Natural History Museum was opened specifically for natural history collections. But Charles Darwin managed to persuade John Edward Gray, who was the keeper of natural history then, and also the trustees of the British Museum, that all of the barnacle collection from the museum would be sent to his home in Down House. And he literally, for 8 years in the 1840s, I think it's 1842 he started those studies, he compulsively, obsessively studied barnacles, so much so that in one or two of the letters, so he's a prolific letter writer, where he documents that his children thought everybody's father studied barnacles, that when they visited their friends, they said, "Where does your father study the barnacles?" So you can imagine how obsessed he was. He was very ill at that time as well, and I know there have been social historians that have studied what he may have been suffering from at that time, whether he was lactose intolerant and so on and so forth. So studied the food that his wife Emma gave him to eat. And he also, it's documented that he did put aside his work on the origin of species at that time as well, although he was very fraught and ill and kind of cursed working on barnacles, but he still, he ordered the best microscope from France so that he could study them. And so the work that I have been doing, you can't believe that from the 1840s, there's still work to do on a collection like this. But there is, in making it digitally accessible, but also you can just about see there are some like paper labels stuck on these boards. So all these barnacles are stuck on. Some are loose because the glue has failed over time. But underneath each board, there are bits of his handwriting and like bits cut out from his notebooks and where he's made comments about the distribution of them, what species they are. So what I am doing, so he did publish 2 volumes of a recent monograph in the 1840s on barnacles, and I am cross-referencing the material, just doing an audit at the same time to see what we have got, and also to designate what he named as type species. So I told you at the beginning, the first of a new species, because they're not actually all marked on these specimens. So there's still always like work to do. And he's a very coded person. Anybody who's studied any of his other material or work that he's been doing, he has written some numbers on the actual, I would say, the head end or the shell, the top part of these stalk barnacles. It's got a B number, and then it's got a sequence. It's got a B, letter B, then a sequence of numbers. I actually thought British, but no, because they're from all over the world. And essentially, he didn't actually collect any of these barnacles. He collected one barnacle. I think he christened it Mr. Anthrobalanus on the Beagle voyage, and that's embedded in a slide at Cambridge Museum of Zoology. It's a very blackened slide, and near enough the specimen is destroyed. So these specimens under my care are from people worldwide, some of them that he wrote to that weren't actually like bird collectors that weren't actually looking for barnacles, but he said, well, if you're anywhere near the coast or somewhere to trade with somebody, can you send the barnacles to the British Museum? So as well as documenting that and verifying various scientific points on type specimens, I'm also making it more accessible so the collection can be digitised, so be handled by other people, doesn't need me to look over them. So I've worked with a conservator at the museum and to get them into various what we call unit trays. And the drawers that they're in were cork-lined, which is acidic, can be acidic to specimens over time, and various pins as well. And so all of the cork of the drawers have been scraped out and then lined with archival paper. And then, as you can see, all of the specimens, some of the boards are cleaned with a smoke sponge. So I've learned how to be remedial conservator as well, to assist in the process of cleaning the specimens and the boards and putting them back. And then they'll be available to be digitised, and that the actual pictures of the specimen and the data will be all available together. The other thing that has changed in museum work, so it's not just looking down a microscope anymore to tell you to look at the characteristics in an animal, do dissections, and to look at printed field guides to tell you how close one animal is related to another or within its own group, its own family. But what you can see here on your, get my left, so right, so it is your left as well. But the type specimen that's marked but red, so this is Nephrops challengerii, I should say. And so that was collected on an expedition called the Challenger. So the ship was called Challenger. And that started in exploration of the world's oceans from 1872, and if I get my dates right, to 1876, I think. On that expedition, 4,500 new species were found to be new to science, I think 250 volumes of scientific work was published. There is still, under my care, a lot of challenger material still for the jars to be opened, to be identified. So again, as I was explaining with the barnacles, there's still a lot of work to do scientifically and curatorially. But where things have changed, you can see that from collecting that in the field, and I must say that the Challenger expedition was officially considered the birth of oceanographic science, but there were naturalists and people doing ocean surveys before that time. But it's where, you know, scientists worldwide came together on board ship to answer particular scientific questions like how deep is the world's oceans. But then leap forward to 150 years later, we've then got this sample of Nephrops norvagicus, and that jar is painted blue. So within my institution, that indicates that it's preserved in 100% molecular grade alcohol or ethanol, and has been used and can be used in future for DNA sequencing. So you can see it says that it's got a museum number, NHM UK, and 2022, and that's when that was collected that year, and it's part of our Darwin Tree of Life project, where we're sequencing a lot of animals and plants, I must say, and getting that DNA code and making that accessible through the database. And so for 150 years, you've gone looking more in depth, and molecular sequencing is, moving at a pace of how we connect the relationships and the evolutionary trees of animals, because sometimes things may not visually look like they're related. And an example are pentostomids, tongue worms. By their name, tongue worms, you would think it's a worm, but it's actually not a worm. It's molecularly, it's closely related to crab, shrimps, and lobsters. So go ahead and check that out when you can. But it's just showing how important, and these molecular techniques are to not do away with traditional taxonomy. It's to complement. It's those sciences and techniques to complement the science for us to know more about the animals of the sun and answer some of the questions they couldn't answer 150 years ago, or never imagined that their samples will be used in molecular techniques. So, digital collections, data and public engagement. Now, this quote actually was great because it came out of The Biologist, which is a magazine by the Royal Society of Biology. And actually the article is about digitising the marine invertebrate collection at the Australian Museum. But I love this quote. It says, "You might imagine digitising museum specimens to be a quick and simple process, but there is more to it than photographing the good side of a lobster." And I thought, gosh, that suits me down to a T. And the picture that I've got down at the bottom is a lobster that I christened Bluey, because it was electric blue. And myself and another colleague, Paul Clark, got a phone call from a fish market, a well-known fish market, Billingsgate Fish Market in London. And they often see interesting coloration in things such as lobsters. So here we've got an electric blue lobster. They did also give us a bright orange one, and what happens there is that the proteins that are to do with colour and binding as a lobster forms and grows, and now I can't remember the stats, but a very very high probability that you get this kind of form of mutation, and it gets electric blue, and the other one a bright orange. We didn't put these, we make a decision not to put these live lobsters into the collection, as they're glad to know. And so they went to, one went to our London Sea Life Centre, and then the other one went to, in the middle of the UK, a place called Birmingham Sea Life Centre there. And so that's where they'll live out their days, and when they pass away, they will come into the museum collections. And that is part of our And so through our collections, there are things that have died naturally in aquarium and so forth, and things that have come in from zoos historically as well that end up in the collections too. So Bluey, still doing good. So I was talking about digitisation, and that has become a major thing across a lot of cultural facilities as well as museums. This is a standard setup. This is just a picture of a setup that I took a picture of. I'm repeating myself there, I just noticed that. When I did a residency at the Natural History Museum in Berlin, and they were actually emptying out all of their bird galleries there. And I had the opportunity of spending a couple of weeks with them and looking at various setups. We have the same in the UK. And actually in Berlin, they had volunteers doing the imaging as well, being trained up to use the camera setups and so forth. But where I work, we do have people that are known as digitisers, and it's their role specifically to do that. I mean, as you can hear, I have a lot of other things to do, and it's quite good to have specialists with that I don't have to be trained with any updated equipment, that they're just people that you can hand it over to and trust them as well to look after the specimens and do that kind of work. And that will be available online. There's also even more technical equipment. So we have CT scanners. We used to, many years ago, use the CT scanners that were available in hospitals for our larger specimens. We no longer have to do that so much because our tech and our equipment at the museum is being upgraded all the time, and we're having bigger and better facilities that we can do that in-house, and that we can offer that opportunity to other institutions as well. So not just for natural history, but for cultural items too. And CT scanning and electron microscopy allow even more minute detail and seeing different features to the microscope that you saw that I was looking through in one of my very first slides. So again, this is moving on, the technology is moving on all the time. But one of the issues is around some of our wet samples, whether you spend the time to take things out of the alcohol and image them, the time that takes, and then the costs involved of person time to do that manoeuvre, or whether we do that through the glass jars, and the jars also distort imagery too. But anything such as the barnacles that I showed, or any dry material, dry coral, can go through the process of going through CT scans and small fragments of specimens. And so I mentioned the barnacles that Darwin looked after, but I also have under my care, aren't I a lucky person, Darwin's corals. So 22 corals that were collected on the Cocos Keeling Islands during his first voyage on the Beagle at the 1830s. And believe it or not, this material, like the barnacle material, is still significant. As some of you may know, Darwin developed his theories and his thoughts around atolls, barrier reefs and lagoons. The specimens themselves that you can see here, some of them are on display to the public in the image on your right. The other one image is with a letter, but the specimens themselves didn't get official identification to species until 2008-9, when I think, correct me if I'm wrong, anybody, but we were commemorating the 150th anniversary of the publication of The Origin of Species. So again, how long it takes, and we don't often have the expertise, the taxonomies. I mean, I was lucky to work with, and I'm still learning from him. I'm still considered a youngster after 30 years in the trade, but coral specialist Brian Rosen, and was able to work with him they also had catalogue numbers from the 1840s that we needed to use a light source that could illuminate the faded ink, and also looking at Darwin's letters and work at the time, the publication on these corals, to actually identify them to species. Some of them just have a genus name, so the first name, as it were, and not fully identified. But that's better, considering that there were no names, just A, B, C, and D, and the locations they were collected from previously. So then that gives us a hook to identify any other specimens that have come after and that from those areas. So still very important and valuable. Something else that is happening, so I mentioned about CT scanning, but around handheld scanners. They may have been used already in some other worldwide collections, but for me, probably about 6 or 7 years ago, had a PhD student that was based in Australia, but wanted to access our historical collections because we have huge time series, 80 years, 100 years worth of environmental data and specimens. And what this chap, Kyle, wanted to do was to use the company's called Creaform, and their handheld scanner that they used in the motor industry and the aviation industry, used that handheld scanner at our off-site store where we house just the dry coral material. And the beauty of the site that we have, that it was bigger than this, the floor space that we could lay out. He was looking at Acropora, which is cosmopolitan and has lots of species, but looking at Acropora of specific species, laying them out and looking at their shape and form and scanning them. The image in the middle does rotate, but I don't think it's going to play for me today of how to do that, and to give an idea of how coral reefs, how their shape and form over time evolves. Must say, though, that the collections are often, when you're doing your field collection, you're being quite biased. You're looking for specific things and shape and form in there. So we had to think about that too. But Carl has just published some work there that's available. I put the link there, but you can also just search that online by putting in ocean with 3D scanning and get that up. But that's new tech that is handheld and is able to come off site, because I said back at the museum we have in situ scanning machines, but it's very difficult and laborious to keep taking specimens back and forward, and also the fragility of the material as well doesn't lend itself to keep going too far too often. In terms of digital discoveries, so this is a web page that I developed with our in-house digital team. And it was around the Chagos Island coral collections. So coral collections, 6,500 of them that were collected on 3 expeditions in the 1970s, just before or at a time when the CITES, that in terms of wildlife trade, banned the collection of corals. You have to get special permits for science, if you're doing any scientific study. But this collection has been coming into its own historically, because Charles and Anne Shepherd, who were part of this expedition, and a famous UK botanist called David Bellamy, they went to the Chagos archipelago collecting this material. And now Charles and Anne, they're now part of the Chagos Conservation Trust, and they are actually coming back and looking at the species identifications that they made of the corals then, that are now in my care, and kind of updating and taking images of the coral skeletons as well to create comparative guides in terms of what the reefs, the living reefs look like now. And as some of you may already know, that the Chagos Archipelago many years ago, now I think it's probably over 10 years, became a marine protected area. But there's still some value of how that historical collections can contribute to the knowledge of the living reefs. And we do, under special permission and form filling, allow for parts of some of the corals to be taken for molecular sampling. And I'm actually going to be working with some people at Oxford University looking at a species called Tenella. So it's kind of a boulder, a circular coral that kind of looks like a brain, essentially, and doing some work there. I think this is just talking about, having partnerships and sharing environmental data, which I think I said. So this is me in our coral store off-site with lots of things around me. But what is the future of digital collections and the data? Well, some animals are quite hard to digitise, such as jellyfish, so we're still working on that. But the data I'm making available through museums' NHM data portal. But if anybody has any ideas that works in museums that has been digitising jellyfish, then let me know. But the other thing that we are working on, and that has been exhibited in the galleries too, and originally the intention of these models was to inspire the public to care about the natural world, to care about the world's oceans that some couldn't necessarily afford to go and see or not privileged enough to go on expedition. And these are... yes, I am showing you the right slide. And so... glass jellyfish and an octopus there. So under my care are a collection of over 187 models, and there are models like this distributed throughout the world, but they were made by a father and son team called Leopold and Rudolf Blaschke. And I don't think they thought that their models will become so well prized, and they're, within the intersection of art and science. So Leopold, the father, his intention originally, before his son Rudolf joined the business, intended that these should be as lifelike and as possible. So you can see there's a certain, or they try to get a certain fluidity or motion through glass. One would say that the glass jellyfish are as fragile as they are in the ocean, the actual things, gelatinous creatures in the ocean. And they are amazing to look at. That's such a work. So the jellyfish were created in the 1870s and the octopus in the 1880s. But I do have sea anemones, so literally the collection ranges from the 1860s right up to the 1890s, before they went on to exclusively make glass flowers for Harvard, essentially, which are part of their botanical teaching collection. And if anybody is in Harvard in Boston, go and see them, because they're absolutely amazing to think that they're made from glass. They tried not to make everything shiny, and so they used ground glass to kind of map the surfaces to make it as lifelike as possible of the animals that they wanted to portray. And so they were used as teaching models around the world, but now shown on display, such as our Treasures Gallery since 2012. So this is where we've used CT scanning to actually care for these models, and artists as well as scientists have been interested in looking at them. And you can see the illuminated bits are the suckers and the other the eyes and the other spiky bits on the head region of the octopus there. But what some of scanning has not only illuminated the character, the shape and form and characters of these models, but also internally how they moulded the glass together, used wire armatures, the kind of glues that they use, and so on and so forth, that can help help us look after these models for, the next 100 years so that they're available for other people to come and look at. The other way that museums are changing in terms of how they portray things such as corals and the underwater world, they've done it for a very long time, but a different way. So this was a mark in the sand, as it were, in 2015, where Natural History Museum in London did an exhibition called Coral Reef Secret Cities Under the Sea. And it was the first time, let's say, in a modern era, that we had more than 250 actual specimens from our collections as part of this exhibition. The tendency can be to just have quite, electronic key touchpad things. They're great, they have their uses, but the other thing is to get more of our material out of the stores. Only 10% of these specimens, there's only 10% of our collections that are actually on display to the public. I mean, we do have behind-the-scenes tours, but it's best to kind of get this material out, to talk about the science that we are doing and to show the unique points of the specimens that we have, and to also create advocates for the planet. To see is to know, and to represent your area, your culture, the world that you come from, and why you should care for these species. So most people thought it was going to be boring just showing corals that look any kind of beige, but it wasn't so, because some of the backgrounds were different colours that can let's say, highlight the structures. So what you can see, so the kind of pinkish organism on top of the coral there are some barnacles, but then you can see some circular projections there, and they are the corallites. They allow scientists to identify to species what a coral is, because they have different shape and form there. And so in terms of the kind of background that we had the corals resting on in the exhibition, actually illuminated a lot more, projected a lot more of these corallites, so people could appreciate, not just for the beauty, but understand how we taxonomically identify the corals. And then at the back of the exhibition, for the very first time in our museum's history, we had an aquarium. And so the aquarium was installed from some fragments of coral from a small local museum in London called the Horniman Museum, where they have an active coral reef studies program, amazing space managed by Jamie Craggs there, the aquarium curator. So they set up the beginnings of an aquarium, and throughout the whole the exhibition period, people can come back, and we also had cameras that were monitoring, and you could see through time lapse of the different corals growing there. And so that was amazing to not only have historical and dead corals, but also then have the living as well. As I said, we've been talking about the environment for many years, and it wasn't until 2021. So the large coral specimen and the sectioned one was, I think, since the 1970s in our impressions gallery, with not much interpretation. It was there for its beauty. And then in 2021, we actually accessioned this material, and I found out why these coral specimens were actually in the museum. And they were part of a study of corals in Phuket in Thailand in the late 1970s near, I think it was a tin, if I can't read it from here, but a mining factory, and where the coral, you see the red band in it, had absorbed that over time while growing. So we have changed the narrative in the gallery about this to for people to understand that industry and certain types of industry can be detrimental and to corals, and look how they can be embedded within the structures, and tell us something about the environment, the past environment, and how we need to do better now. So this is the data portal that I have to deal with, and these are just some provocations, really. I've been learning a lot while I was here, especially at a conference I was at in Auckland last week. How do we do? I work in a museum in the West. How do we deal with indigenous names, putting those into the database as well, having access to those, and keeping other information private and respectful to, such as the Maori community. And not just putting those names within a common names field, having equal authority. Who should input the data? Where do the standards come from for data input? Knowledge base as well alongside that. Indigenous knowledge, oral histories and publications within the database, not to dump everything into a notes field that's not necessarily available to the public, or maybe somebody in another department to me as well that can't access that. So have a more holistic approach, and so that we can have different users to our science if we want people to understand it as well. The thing about open access and benefit sharing as well, environmental data, the issues that are around that, the ethics. So those are all the questions that I have to deal with on a daily basis, talking to my scientific colleagues worldwide, and also it's for the public to understand the issues that we're dealing with as well, and to join in the conversation. And I might be running low on time, but coming forward now to not just my original curatorial practice, but in the last couple of years it's been highlighted, at least since 2020 a lot more, but people were doing this work before me, is looking at collections with a different lens. So these are within my natural history collections, they are considered specimens. They are corals growing on a bottle and a jug. But if you dig deeper, where were the corals purchased from? They're purchased from, you can just about see it, the colonial and Indian exhibition in 1886. 4 million people attended that exhibition in South Kensington in London. There are scanned documents to do with that. It was literally a bit like a world trade fair. So the broken bottle is interesting because it came from that fair. The jug actually was dived up in 1860 from Port Royal in Jamaica. And I've had some artists and scientists interested not only in the coral but in the jug from the United States, where they're going to do an exhibition around this. But also one can only imagine what was going on in the 1860s in Port Royal in terms of the people that were there at that point in time. But these kind of, insights, I'm doing investigations, but there are other people across the world that may have more insight than myself. What was the jug made of? What was it used for? The style of it as well. And so I've been digging deep. So these are all the websites. So again, it's not only scientific websites that I need to look in, but also antiques and so on and so forth. The Wellcome Collection as well, based in London, that give a lot of valid information there. I've been doing a lot of work around whose voice are listened to in terms of colonial legacy of museum collections, and with a marine link. So I was at the Maritime Museum in 2016, where they opened an exhibition about finding HMS Terror that was lost since 1848. And I had the honour of meeting this chap, Sammy Cogvig. And I've got two quotes here that came out of the press and the media. Sammy's a very shy person, and I coincidentally met somebody that knew him who's from the Inuit community as well just recently at the museum, and we were discussing this whole thing. But I'll just read the quotes. The community knew about this for many years, many years. It's hard for people to stop and actually listen, especially people from the south. And then the other quote is, we are in the backyard of the Inuit, so we should listen and learn as much as we can. So within Sammy's community, and Sammy himself had been seeing the exposure, the ice caps melting in terms of revealing the mast of HMS Terror. The story would go around the community. Sammy himself had lost some images that he'd taken of himself with the mast of the ship and didn't think anybody would believe him. But then he got an opportunity to tell this story. And he, along with some people, I think from Canada and the United States, then were in search of the ship and found it. But what I'm trying to say here is about listening to other communities that may not consider themselves to be scientists or whatever, but have specific knowledge, environmental knowledge of what they're seeing in their areas around them, and that we must take note of that too. So where exhibitions are moving forward and changing, and as I said, and Sammy and his community having a voice and being acknowledged. Here at the museum in 2021, we opened an exhibition during the pandemic, so it was opened in three instalments. It's Our Broken Planet, and it's not all doom and gloom because it's how we got here and ways to fix it. And so there are many voices within this exhibition. You can see a picture of me there talking about whether because of jellyfish blooms and overfishing, which is causing that, natural predators for jellyfish larvae are being overfished out of the water, out of the world's oceans, whether there be increased need to eat jellyfish. They eat jellyfish in certain parts of the world. I've never had jellyfish soup. I can't imagine it'd be very tasteful. You'd have to add a lot of other things to it. But, it's to get us thinking about this, of what we are doing and the impact on other populations of other sea creatures, that we have to be mindful of that. So the exhibition is currently closed. Part of it is touring, but we will reopen at the Natural History Museum in 2025. And just lastly, going on to voices. So I belong to a group called Climate Reframe. I was part of the first 100 as we went into a pandemic, to give those black and brown voices as people that work in the environmental and scientific community in the UK, to give them a voice in terms of social justice. I noticed there is, a lot around, justice for land and the Maori communities here, and that is justice in terms of UK voices, trying to help what happens further afield across the world, because I'm of Caribbean heritage, even though my parents met in the UK. But it's about me knowing and other people knowing that what I do in the UK can affect people worldwide. The weather systems that we experience, extreme weather systems, and the extinction or potential extinction of plants and animals in the natural world. So I work with a lot of communities outside of London in the rest of the UK, and this is one science festival. So telling those marginalised communities, what it is to be a scientist, and to inspire them with science. And then the museum itself has young people from across the world that are working with us, and also last year took over our museum, but it's a work in progress with them. And so we're creating advocates for the planet under our Generation Hope scheme as well. And I'd just like to thank you all for listening.